The pre-rRNA promoter DNA in the yeast consists of two regulatory elements. They are upstream element and core element. The upstream element extends roughly from -155 to -60. The upstream element stimulates in vitro RNA Pol I transcription tenfolds. The core element extends roughly from -40 to +5. It is essential for Pol I transcription. At the beginning of assembly of a fully active yeast Pol I transcription initiation complex, a hexameric upstream activating factor or UAF binds to the upstream element. Out of 6 subunits, histone proteins like H3 and H4 are the 2 subunits of UAF. H3 and H4 probably participate in DNA binding. Trimeric core factor or CF along with TBP then binds to the core element. The TBP makes contact with both the bound UAF and the core factor. Finally, a complex of RNA Pol I and a monomeric factor called as rRN3P associates with the bound protein. The RNA Pol I is positioned near the start site. With this, a fully active yeast Pol I transcription initiation complex is formed. Please like, subscribe and share.